I read more and more papers from Rob Russell, who was at the time a group leader at EMBL and worked with computational methods on protein structure comparison to find similarities between protein structures that were indicative of shared function. And that I thought was totally fascinating, to basically find small local similarities that are shared between otherwise entirely unrelated proteins, and where these similarities were then also indicative of a shared function. For example, the most famous example, at least at the time, were the chymotrypsin and cytolysine proteases, so meaning proteins that can cleave other proteins and do this via very similar mechanisms despite being otherwise unrelated. I thought this was cool.
It must have been very difficult to do such a job, in the era pre-AlphaFold with, I guess, not so many structure to compare to. So how was it, and how do you think these new methods impacted that kind of job?
Yes, it was indeed much more limited than nowadays. So we worked off experimentally characterized structures only. But fun fact, we were indeed as a group, because Rob is very well known and established in that field of protein structure analysis and also the computational protein folding or protein fold prediction. We were for 1 year the judges of this famous CASP competition. Oh. And at the time, while there was already progress visible, it was of course not at the level where we are now with AlphaFold.
So the CASP competition was actually the benchmark to develop AlphaFold. So it must have been exciting to be part of that. I think it's a pretty unique example of a field that had, everything in place for actually developing and deploying the promise of, I guess, computational biology for that particular aspect, right?
Yes, so the CASP competition was already established and running for quite a few years when we were asked to be judges, or Rob was asked, and then we did it as a team effort. So that was a quite interesting experience. So the process that allows every one of our cells in our body that carry the same genome, the same genetic information, and utilize this or express it in very different ways, such that a cell that you find in your liver will have very different properties than a cell in your brain or in your bloodstream. And I was totally fascinated how our cells can take that same genome, read it, and utilize it differentially. Express certain genes only in liver, where they might aid metabolism, and other genes in the neuron, where they might aid neurotransmission. That process I find entirely fascinating, and in particular because it is kind of written in the DNA in a code that we didn't understand at the time, and we don't really still understand. And as I mentioned in the beginning, I was always fascinated by this interface of biology and computation, and this digital information where evolution found a way to not only write down protein sequences in a digital way in the DNA, but also when and where these proteins need to be activated or expressed also in the same DNA, but in a very different code or language. And that is something that started there and still fascinates me. So we're still working on this problem. Now, 25 years later, almost. And I think it's still one of the coolest problems. Now, particularly with the help of AI, we are for the first time able, as a community now, to really kind of computationally find such regions in the genome and predict where this region might switch on or off gene expression. And we also can now, again, as a community, use these AI tools to build synthetic sequences computationally designed that then also drive expression. I think this is totally fascinating that we can really go within these 25 years from not really understanding this language at all to now being able to write it out on our computers and then really have these sequences work in our cells.
Give me a sense, in the last maybe 25 years, right, of how technology helped you, make this picture emerge, because the computational technology must have changed a lot. I mean, certainly we were not talking about AI 25 years ago, right?
No, I think 25 years ago there was, of course, already a notion of machine learning where one could use, let's say, at least from today's perspective, relatively simple models to establish associations between certain types of data and other types of data. Right? For example, one could already, of course, at the time, look at certain, let's call them words or motifs in the DNA, and then associate their occurrence or abundance with certain functional outcomes. Right? That was already done at the time using models like regression or support vector machines, which I think people term machine learning in the broadest sense. But then only much later, I think in biology it started probably 10 years ago. David Kelly was one of the first who applied what we now call deep learning with convolutional neural networks to genomics. And since then, many groups, including ours, picked up these technologies. So we can now, with these more advanced computational models that people now term AI for artificial intelligence, establish these functional relationships between DNA sequences and functional outcomes, right? Be it the binding of certain transcription factor proteins to the genome, be it what people call accessibility of the DNA within chromatin, right? Where basically chromatin can either compact DNA, make it inaccessible, or loosen up and make that DNA accessible to regulatory factors. And also what we work on is the regulatory activity, right? So all of these layers can nowadays be predicted using these convolutional neural networks.
Now that, of course, technology progresses, is it, worthwhile actually repeating experiments that perhaps you have done 20 years ago with new technology? And, do we get useful information by taking that kind of approach?
Yes, I think so. And I mean, one of the clearest examples, again, now from our field of gene regulation is, for example, comparing classical genetics where proteins are depleted by mutagenizing the DNA versus a technology that actually came up during my PhD times, RNAi, where gene expression was perturbed at the RNA level. And now we can, of course, also since many years already, at least maybe a decade or so, we can now perturb proteins directly at the protein level, with either chemical inhibition or what we now very frequently do in the field is degron-based rapid protein degradation. And it's really interesting how basically having this gain in temporal resolution and being able to work with much more acute perturbations where we now learn very new aspects, right? Where we can look at the direct functions and roles of certain proteins in gene regulation.
Yeah, and then also compare differences with
Exactly.
the other types of perturbations. I mean, this is super exciting.